Ribosomes are molecular machines found in all living cells that play a crucial role in the synthesis of proteins. They are made up of RNA and protein and are responsible for translating genetic information encoded in messenger RNA or mRNA into proteins. In this explanation, we will discuss the structure, function, and significance of ribosomes in depth. Structure. Ribosomes are composed of two subunits, a large subunit and a small subunit, each made up of RNA and protein molecules. The small subunit is responsible for binding to the mRNA molecule, while the large subunit is responsible for catalyzing the formation of peptide bonds between amino acids, which are the building blocks of proteins. Ribosomes can exist in two different forms: free ribosomes, which are not attached to any membrane, and membrane-bound ribosomes, which are attached to the rough endoplasmic reticulum in eukaryotic cells. Function. The main function of ribosomes is to synthesize proteins, which are essential for the growth, development, and maintenance of all living organisms. Ribosomes do this by reading the genetic information encoded in mRNA molecules and using this information to build a chain of amino acids that will ultimately form a protein. The process of protein synthesis is divided into three main stages: initiation, elongation, and termination. During the initiation stage, the ribosome binds to the mRNA molecule and identifies the start codon, which signals the beginning of the protein coding sequence. During the elongation stage, the ribosome moves along the mRNA molecule, adding amino acids one at a time to the growing protein chain. Finally, during the termination stage, the ribosome reaches a stop codon, which signals the end of the protein coding sequence and the release of the newly synthesized protein. Ribosomes are essential for the survival and function of all living cells. Proteins are involved in a wide range of biological processes, including DNA replication, cell signaling, and metabolism. The synthesis of proteins by ribosomes is a critical step in these processes. Mutations in the genes that encode ribosomal proteins or defects in the ribosome itself can lead to a range of disorders and diseases, including Diamond-Blackfan anemia, congenital heart disease, and various forms of cancer. Basically, ribosomes are complex molecular machines that play a critical role in the synthesis of proteins. They are composed of RNA and protein and exist in two different forms: free and membrane-bound. The main function of ribosomes is to synthesize proteins by reading the genetic information encoded in mRNA molecules and building a chain of amino acids that will ultimately form a protein. The significance of ribosomes is highlighted by the fact that mutations or defects in the ribosome can lead to a range of disorders and diseases.